To welcome Dr. Ryan Shum. So Ryan has been working with me since November 2024. It feels like longer, but in the absolutely the best way, on the Renewing Biodiversity project, where we're studying together how people collaborate across disciplines in a really, really large conservation project, and what happens when environmental scientists start to put people at the heart of these questions around conservation and biodiversity renewal. But today Ryan is going to be talking to us about his PhD work. So he's completed his PhD in November 2024. His PhD was entitled Caring for Microplastics, a multi-sited laboratory ethnography, and he completed that in the geography department here. That thesis received the UK Assist, which is Association for Social Studies of Innovation and Technology, I think, PhD prize last year. And today Ryan is not going to try and present the entire thesis, but instead actually is going to be presenting a paper in progress that he's writing at the moment, which he can tell us a bit more about the kind of input he'd like on that. So over to you, Ryan.
Thanks, everyone. Thank you for being here. And yes, as Angela said, this is a paper in progress. Really welcome any comments, questions, just even of how the paper progresses. And I'm really close to kind of having a draft out. So this is kind of where it is at the moment. And apologies in advance that there's going to be some reading involved. I'm going to try and keep it a bit more exciting than just me on a page. So here we go. So the paper is titled Sensing Ubiquity, Encountering the Geographies of Microplastic Science in the Laboratory. And obviously in recent years, microplastics have become a hugely topical environmental issue in the UK and globally. The headlines go something like this: microplastics everywhere there, exposure to plastics and plastic chemicals are toxic to humans, more than human life. This talk kind of tries to take a step back and examine the laboratory processes and practices that enable this ubiquity of microplastics to become sensible. And some of these are some of the open questions that I'm trying to think with today here. So how do you study something that is imperceptible and everywhere all at once? What does that look and feel like? How is scientific knowledge produced when the boundaries between sample and contamination become ontologically indistinguishable? What are the geographies of this chemomaterial ubiquity? So this paper is an account of how microplastics come to matter in the lab, how researchers contend with a multitude of plastics that circulate through these spaces and processes, but also how scientific practices and technologies are mobilized to respond to this material pervasiveness. So a little note on the image that's on the background of this slide. You can encounter microplastics outside of the lab. So these petri dishes were designed by one of my interlocutors for use in a public workshop. Each are meant to kind of represent a different environment where you might find microplastics. I'll come back to this in a bit, so I'll just move on now. Okay, so it's a bit tricky to know where to start with the presentation of microplastics. They're not just materially, but discursively ubiquitous. Kind of set the scene at a few quotes. In an opener to a paper on environmental estrogens, Celia Roberts writes, "There is something suspicious in the water, in the air, and in the ground that is producing change in human and non-human biological systems." Writing about the herbicide glyphosate, Warner et al. Talk about the ubiquity of chemicals demanding new ways of thinking about human nature assemblages and their boundaries. In a recent article, Steve Hinchcliffe writes that objects of STS and environmental geography are not what they were. So some transformations to bodies and environments have been very visible, acute and felt. Others, for example, like the Great Pacific Garbage Patch, which is the image in the slide, sort of an area infamous for being the largest accumulation of ocean plastics, is not something that can be found. Its materiality falls beyond the threshold of human perception. So I don't really have time for a sort of comprehensive history of the emergence of microplastics and microplastic science. But I think it's worth noting that something has changed in the last 20 years of plastic research and discourse on plastic pollution. So in 2004, Richard Thompson and his colleagues, man pictured in the top right, published a landmark paper titled "Lost at Sea, Where Is All the Plastic?" Most famous kind of finding of that paper is where the term microplastics was coined. And as the title of the paper would suggest, marine biologists working on plastics at the time, there weren't that many, were confronted with the problem of missing plastics. So the quantity sampled and modeled using data from sea surface trawls did not match the exponential year-on-year increase in plastic production and known quantities of plastic waste entering the oceans. 20 years later, the question is no longer where is all the plastic? And this is Richard Thompson himself again, penning an article in The Conversation with the title, "Everywhere we have looked, we have found evidence." Recognition of the toxicity and harm from plastic exposure and associated plastic chemicals on human and ecological health is widespread, even if still contentious. In the latest mapping, there are over 16,000 plastic chemicals known to be associated with the production of plastics, with 4,200 known to be persistent, bioaccumulative, mobile, and toxic. I mean, these numbers are not particularly exciting or interesting by themselves, but they say something about what has been made public and what has been tested in the lab more than anything else. And of course, the scale of the problem has prompted the UN Environment Assembly to adopt Resolution 514 in 2022, beginning the negotiations for an international legally binding treaty to end plastic pollution. A process that has stalled at this point, but here we are. And a space where microplastics are especially ubiquitous in the UK has been the Guardian news publication. From the headlines, at least. I mean, this is not sort of discourse analysis or anything, but the very nature of plastic pollution seems to have changed. Geographically, we're no longer constrained to the oceans. Nowhere, nothing seems to be uncontaminated. They have been found in human reproductive systems, in the brain, in the wilderness. And these sorts of news publications arguably contributed to this sort of alarmist, perhaps, discourse around microplastics. And within this recent constellation of interest, The Guardian also published a set of exposés shining light on the methodological challenges surrounding the detection of microplastics on human tissue samples. So this is the first one that says, "Bombshell casting doubt on the discovery of microplastics throughout the human body." And on the surface, they seem rather shocking. So the article draws attention to false positives, errors, misinterpreted results surrounding certain high-profile studies that were widely reported by the news media, including The Guardian. And to sort of talk through this briefly, the concerns were largely around a paper on the detection of microplastics in human brain samples. The concerns were surrounding the analytical method of choice and the lack of quality control protocols in place. And specifically, how lipids in the brain, when pyrolyzed, can also mimic polyethylene, registering false positives. And in the following editorial, and these are some quotes that I've taken from that editorial on the right, they write that this is not an attempt to erode trust in science, but simply how science operates. So I think particularly something that's particularly interesting, I think, from the quotes. It's unfortunate that scientists working in this field did not show more caution. There was something about how medical researchers entering this field didn't quite understand the quality control and the rigor and technical expertise in chemistry that were needed to work with microplastics. Perhaps until more clear and widely agreed standards are established, that great care should be taken before results are published and reported in the media, something that the Guardian themselves have been very much complicit with. So I think there's a lot more to unpack here, not also thinking about industry interference, production of ignorance, but I guess suffice to say, now is a good time to pay critical attention to the epistemic practices of microplastic science. It is timely to investigate how microplastics become sensible, how this ubiquity is evidenced and enacted, and what the challenges working with microplastic materiality are in the lab. So this is the plan for today. The next couple of slides will turn to microplastic materiality. It asks what kinds of matter are microplastics. I've approached the question in two ways, and the purpose of this will hopefully become apparent shortly. And I think understanding, or having at least a basic understanding of what kinds of matter microplastics are, is crucial for the empirical narrative of the next. Then I move on to the question of how we know what we know about microplastics. Here I introduced a conceptual framework of sensing as a way of characterizing laboratory practices and offer some background on the emerging field of microplastic science in the UK and the field work that has informed this work. Loosely centered on the scientific processes of sample extraction and identification, contamination control, and chemical characterization. I will then talk through the spaces, practices, and technologies of sensing that are assembled in the lab to make microplastics sensible. These empirical cuts following Barad show how these sensing practices interface and contend this indeterminate materiality to the enactment of partial momentary disentanglements. So this is a conventional introduction you might expect when to talk about microplastics. We begin with definition. Widely defined as a solid plastic particles within 1 micron to 5 millimeters in size. So we're dealing with a product of fragmentation. Some microplastics are termed primary microplastics, which are plastics manufactured within this size range. So examples include things like nurdles, which are pre-production pellets, biobeads, which are used in wastewater treatment centers. But the vast majority of microplastics encountered are secondary microplastics. They form from the breakup of larger plastic objects. Synthetic polymers are largely resistant to biodegradation processes, not physical or photodegradation. So rather than breaking down to their constituent parts, they break up into smaller and smaller pieces. All plastics are composed of polymers, functional additives, as well as other unintentional and intentionally added chemicals. In this sense, we're not dealing with one microplastic, i.e. many of the same thing everywhere, but a multitude of chemical and material variation and complexity. So the classic kind of quote that comes from the scientific literature is this one in green. So the idea is that microplastics are not microplastics, they are a class of contaminants, just like pesticides are not pesticides, are not pesticides. So at the upper limit, microplastics are readily amenable to the human senses. If you swallow them, you might spit them out. But particles at the lower limit and nanoplastics have demonstrated the capacity to translocate across cell walls. The impacts are chronic, slow, transgenerational, which means they are harder to detect in laboratory studies, and also unequal, impacts some communities more than others. Well, microplastic matter version 2. So I think you can introduce microplastics as a scientific issue with scientific facts that are each more anxiety inducing than the other. But I think we might also think about microplastics in a language that are a bit more meaningful for people in the room. So what can be said about the materiality of microplastics? Microplastics seem to epitomize this processual quality of materiality. They're inextricably bound up in the process of breaking up. If you sample now, and then 5 years later, with no additional inputs, you would have a greater number of microplastics. Plastics also hold in their namesake a quality of plasticity, this capacity to take and receive form. But at the same time, the refusal of plastic materiality to break down might also expose its material limits. Media scholar Heather Davis writes that the plasticity that plastic embodies is epistemic rather than necessarily ontological, i.e. there is not actually much plasticity in actual plastic objects. They are also relational. They're inextricably enmeshed across the biochemical processes of modern human life and metabolisms. Microplastics are always encountered in messy entanglements, confusing the blurry boundaries of body and environment. This relationality is one of the primary reasons why sensing is so challenging. The propensity of microplastics to move across body boundaries, evade detection, gather and concentrate environmental contaminants, show that they're far from inert objects waiting patiently for humans to find and manage. I've just put agential down here, but this is not necessarily this is not a celebration of their vitality or a romanticization of their agency, but perhaps this is a recognition that they have a capacity to affect and be affected. And following Braun and Watmore, thinking with agency here might be about attending to the specificities that are important to the material relations enacted by microplastics. And maybe this is not also so different from Lapowski and LeBron's definition of scale as the relationships that matter to defining an issue. And this is more of a general point here at the bottom, which seems to be hard to get away from this language of process and entanglements when you're thinking about these materials. But I would argue that maybe it's not actually also so different to the descriptions of ubiquity that are told in the scientific literature. So how do we know about microplastics? Suffice to say, much of this research remains largely an endeavor of the natural and biological sciences. To generate data on microplastic requires sampling regimes, expensive instruments, lab chemicals, analytical capabilities and expertise. This obviously doesn't only occur in a lab, but having a lab helps. Developing ways of sensing pollution were crucial to responding to, acting on and with, and caring for the environment. Although not a panacea, generating data and evidence of pollution and contamination is a key strategy for publics to demand action, change, and justice. But at the same time, perhaps it is not so surprising that across the social science literature on plastics, microplastics are not often the focus of analysis. Kind of like their residual materiality, they sit in the background of the literature, not quite in focus, but never absent. Although I wouldn't call this a problem or necessarily even a research gap per se, I think there is a risk that these forms of plastic matter become reinscribed only as unfortunate side effects of plastic use, the consequence of bad management, or reified as chemical bad actors. Microplastics have seemed to become this contextual background on which the timeliness and urgency of plastic research is demonstrated. And of course, as ever, there is a rich seam of scholarship that explores the plasticity of plastics that this paper builds on. A non-exhaustive list here is pictured on the screen. This body of work does not necessarily center the study of microplastics, but emphasizes the processual nature of plastic pollution and situates their analysis across the porous boundaries of the chemical material, body, and environment. These authors, working broadly across STS and media studies, rethink plastics through an attention to the boundary transgressions enacted by plastic matter, is situated in ground feminist, queer, and anti-colonial commitments. So the intangibility of microplastics does not necessarily lend itself to conventional qualitative research. But as Shapiro and Courtesy write, there are ways in which ethnographers can study chemicals. And it is the sensing technologies and collaborations that are the focus here today. So the reason for shifting from thinking about detection to sensing in microplastics is conceptually driven, but it is also pragmatic. Initially, I was confused by the ways in which monitoring as a practice was being deployed in the scientific literature. So I understood detecting and monitoring as a recursive, continual relationship with a particular site or body that measured change through time. As things change, while things stay the same. And yet the practices I actually encountered in the lab and during my fieldwork that are described also here are largely examples of snapshot sampling of a specific site or body, i.e. rigorous enough to give us an indication of the presence or absence of microplastic contamination, but only speak of a particular time and space. I think there is also an imaginary associated with detection practices as sets of procedural, prescriptive, disembodied, and automated processes that are universally applicable and standardized. I've put an image of a carbon monoxide detector here as kind of a straw man maybe, but I guess the point is that sensing microplastics involves a whole series of things coming together, in the lab in order to make them matter. And the conceptual language of sensing enables us to focus on the materiality of microplastics, how they interact, confuse and disrupt knowledge-making practices. But it's also a recognition that following genetic discoveries, sensing and sensory experiences of microplastics are decidedly more than human. They involve a wide cast of entities and ensembles of sensing, including and extending beyond human bodies, technologies, and also environments, which come together to make microplastic matter sensible. And it seems like we're not just dealing with a proliferation of sensors and sensing technologies, but this ubiquity means that everywhere and everybody has the potential to become a sampleable subject. So a few words on the field of microplastic science and also the field work that has contributed to this paper. There is a geography to microplastic science. The vast majority of labs equipped to sense this contamination are located in the global south, yet the impact of microplastic exposure is uneven. The UK itself is a key site of microplastic science knowledge production, and many of the key actors of the field are based here in the southwest. So on the top here, we have 3 scholars who together won the Volvo Environmental Prize for raising awareness for plastic pollution. Richard Thompson is based in Plymouth, Maura Galloway here in Exeter, and Penelope Lindquay in Plymouth Marine Labs. So microplastics is very much a field that was born in the southwest of England. And so hence why the fieldwork was very much structured around the UK. As I mentioned, the term was coined in 2004, but scientific interest was slow. It wasn't really until 2017 that publications would be going by that bibliometric count. Publications passed 10 publications per year. My fieldwork was conducted between 2021 and 2022. So this was in and out of the COVID lockdowns. So we had to be very creative about the ethnography that was possible at that time. But the material that I'll talk through today is based on 31 interviews, constant observation in 2 labs, conference ethnographies, including microplastic conferences, going to the plastics treaty negotiations, and lots of document analysis of the plastic literature. And just to say that the empirical focus today is on presence and absence studies. So these are studies that have looked for the presence or absence of microplastics. And so this is only a snapshot of the field's practices at a particular time and not representative of those. So the following material is a composite narrative of the sorts of laboratory practices I encountered in the field. It roughly follows the flow of experimental processes deployed to analyze microplastic contamination in spaces like lake sediments, sewage effluent within marine biota, but it is not a step-by-step guide to analysis. I use cuts as a structuring device for a few reasons. Firstly, to emphasize the literal separations taking place between what is plastic and the environment, what is plastic and non-plastic, sample and contamination, production of data and human bias, but also the difficulties of disentangling these blurry boundaries. But it is also to think with Karen Barrad's notion of agential cuts, which understands the epistemic practices and measuring apparatus that come together to enact specific material realities as inseparable and implicated in the realities it produces. They suggest that scientific practices are always participating in the materialization of the phenomena that they study. And in particular, I'm also interested in the role of exclusions that Holland et al. And others have identified as key to Barrad's theory of agential realism. Agential cuts resolve uncertainty and indeterminacy in particular ways and not others. And similarly, Michelle Murphy writes the regimes of perceptibility that govern scientific practices. Any way of materializing contamination as sensible also sets the terms of what becomes insensible. So to rework Kim de Wolf's observation here, Green, I hope that by the end of the talk, we won't be troubled by the need to separate in order to understand connection. And to go one further, I would contend that following these moments of fraught and partial disentanglement is key to learning about how to respond and live with a form of plastic matter that is not going away, no matter how much we want it to. Getting there. So this is one, separating plastics from its entanglements. The first step of lab work involves separating suspected plastic material from the environmental matrix. Essentially, this is all the stuff that you want to get rid of in order to understand suspected plastics. Plastics are solid, mainly insoluble, meaning that to sense, you need to literally separate plastics from their chemical material entanglements. And as one researcher put me, you need to adjust the methodology according to your samples because they're unique, always different from anyone else. They're obviously here not referring to a sense of ownership of the samples, but that need to be attentive to geography of the collected samples. The roadmap here, which I'm sure you won't really be able to see, is to give you a sense of the decisions that a researcher might need to take depending on what environment, what organism that you're working with in order to extract. Just because something is everywhere doesn't mean that the same methods can be used to understand them. To disentangle, researchers have to attune to the interconnections between matter, bodies, and environments. For one of my interlocutors whose research was concerned with plastic contamination in lake sediments, the process of extracting microplastics was not very straightforward because in nutrient-rich sediments, there are many other than plastic particles within the same size range that one has to contend with. So not only do you have to be able to remove and separate the organic and mineral components in the sample, but also ensure that the chosen extraction method doesn't degrade or introduce new microplastics into the experimental system. So making sense of new samples seems like an inherently exploratory process that involves trial and error and improvisation. And for many of the scientists I interviewed, the development of the method is able to sense microplastics in their particular area of interest was as important as being able to establish the presence of microplastics. So after sample extraction, you're still left with lots of stuff. So the next step is the visual identification under the microscope. This process is a pre-selection for the chemical characterization that follows. Earlier work, especially research that looked at plastics towards a larger size range, let's say like more than 500 microns, would rely on solely visual identification alone. And I have drawn on Clay Labs resources, which is Max Lebrun's lab that they lead, as most visual guides are produced by citizen scientists rather than nice pretty images for to use in presentations. So learning to identify what is plastic and what is not involves time. You might work through the filter systematically, grid by grid, line by line, looking for unnatural colors. So your greens, your reds, your kind of blues, as a researcher says, something that pops up to you and says, ah, that's plastic. Sand minerals audibly crunch, like breaking glass, while plastics are often a bit more pliable because of the plasticizers within them. There are some routine tests for plasticity deployed, including a flotation test, or a hot needle technique, which involves touching an object with the point of a heated needle. If an object is plastic, it will react to the heat by melting and bending, but organic materials won't. But these checks for plastics only complement, but do not replace the researcher's eye for plastics developed through hours of microscope time. And actually, in practice, researchers rely on all their physiological senses. It's not just visual identification. The researcher listens, feels through the forceps, might even snap materials in half so they don't lose it to the environment when it invariably pings up to stage. They also draw on their tacit knowledge of the situated material geographies of the samples themselves. So a quote here from Max Lebrun, they say that they're fluent in green threads and the yellow paint chips of the coastline that they work in, but when they move somewhere else, they wonder if the red thing is plastic or coral, if the black round thing is coal ash or plastic or something else entirely. And we return to the sort of mobile lab that I started the presentation with. The idea for this workshop that my interlocutor created was to invite participants to become microplastic scientists to extract particular material from each of these simulated environments. So starting on the left, you have lots of white circles, basically beads. They are nurdles, which are pre-production pellets. So this might be indicative of something like a container spills. The second one is mainly of biobeads, which are these black dots, which are used in wastewater treatment centers as they support the biological breakdown of sewage effluent. The third one is a mixture of fragments, which is what you might more commonly find in sediments. The last one is, I don't know literally, or a representation of nanoplastics, hence nothing you can see really. So different plastic species in different spaces. Okay. So in the first cut, we encountered sensing practices that attempt to separate plastics from other matter. Bodily ways of attuning to plastic matter also. But what is the difference between suspected plastic samples, because we don't know yet, and plastic contamination? Contamination is also a slightly strange, deployed slightly strangely in the scientific literature, in the sense that they're both a matter of concern, what scientists are interested in, but unanimously recognized by scientists as the greatest challenge to their work. You simply cannot count without disentangling background contamination and the contamination that I suppose you're interested in. Microplastics are everywhere. I think we're all clear on that right now. This means that at every stage and at every moment, they hold potential to interfere with the experimental setup. How do you know if the object you're looking at under the microscope originated from your sampling regime? It could have been released from your jumper. Maybe it's a product of atmospheric deposition. I mean, after all, it is all contamination. So this matter that is not supposed to matter comes to really matter in experimental processes. And the laboratory walls, the doors, the fume hoods, and the air filtration systems don't seem to have enabled microplastic scientists to gain exquisite control over the objects of their analysis. Maybe this is a very literal illustration of how acts of measurement, doing an experiment, are also implicated in the realities or results that it purportedly stands separate from. And of course, just like our everyday lives, plastics are likewise essential partners in the lab, all disciplines, and the properties that make them desirable and useful in everyday life also mean that they are probably quite valuable and useful in the lab. So we have gloves that keep researchers safe, masks that were the functions of pandemic life, and when I was shadowing researchers in the lab, to ironically Ziploc bags, which were the easiest ways of transporting material from the field to the lab. And in our own institution's Sustainable Labs portal, they cite a paper from 2015, basically saying that environmental research can be fundamentally unsustainable. I mean, it contributes 1.8% of global plastic waste per year. Each bench scientist in the biosciences lab this is within the same publication, but they nicely put that out. Each bench scientist in the department generated around 1 ton of plastic each in their work. So not necessarily trying to attribute blame or anything here, but that to point out the need to rely on plastics in order to generate evidence of plastic contamination is an inevitable yet ironic part of working with microplastics. There's that conceptual notion of entanglement here, but also a very real and practical inseparability. And this is something that weighs heavily on my interlocutors. And this is just a quote sort of suggestive of this. And moreover, the scientific need is often prioritized over environmental sustainability for using less plastics in plastic work. Using fewer plastics is a way of kind of quality control, but it's never justified in terms of wastage or sustainability in these spaces. And it's because of how plastics confuse experimental results instead. So even if discursively ubiquity often lead us to an impasse, i.e. it's everywhere, so there's nothing really we can do about it. This does not mean that generating data or indeed responding becomes impossible. There are practical ways of enacting distinctions. Quality control, and this is kind of boring, but is essential in ensuring the accuracy and reliability of data generated. Without quality control measures, the results of monitoring may be unreliable, making it difficult to assess, if at all, the level of pollution in the environment. So these thick shades of ubiquity are ordinarily cast as procedural risks to the production of scientific data and a challenge to overcome. But I would suggest in practice, researchers seem to begin from a recognition that contamination is ever present and inextricable. And the use of blanks is just one of these mundane, everyday ways in which researchers can subtract ambient contamination from their results. Cut number 3. So provided everything goes to plan, you've extracted your samples, you've controlled the contamination, you've spent your time manually marking off each fiber or speck on the filter you want to investigate, you kind of run into another problem. So how do you actually ensure the particles you've identified as plastic are really plastic? I mean, it's not possible to infer the chemical properties of a sample via visual methods or imagery alone. How do you transcend the sensory limitations of being human? The device on the right, the Fourier transform infrared spectroscopy, spectrometer even, sorry, might be the solution to your problems. So IR spectroscopy promises ease, speed, efficiency, and accuracy. I mean, as the tagline goes, the breakthrough in simplicity beyond automation. So the aim, as one of the researchers put it to me, is to replace the sensing human and its biases with apparatus such as spectrometer. And what's more, as the field has progressed, visual identification alone no longer meets the contemporary publication standards of the field. In an opening editorial of the journal Microplastics and Nanoplastics, one of the first solely focused on micro nanoplastic research, Bart Coleman's, its editor-in-chief, writes of the sorts of high-quality papers they want to accept. And one of the things he says is that the use of only visual inspection to identify microplastic particles should be avoided. To give you an illustration of the sort of problem we're thinking about, these are suspected microplastics extracted from a freshwater sample. All fibers would pass the criteria that we talked about earlier about the visual identification of microplastics. So they're all largely homogeneous in thickness and in color. They're shiny, don't seem to show any cellular structure. So which samples would you identify as plastic? Which samples would you then confirm through infrared spectroscopy? Keep in mind that in a well-known review of identification methods, they observe that up to 70% of particles that visually resemble microplastics are not confirmed as plastic by IR spectroscopy. So this identification process is time-consuming, requires an experienced researcher able to make informed but always subjective decisions about what to count and what not to count in a sample. This results in inter and intra research variation and is a challenge that is exacerbated by the limited reporting on the different criteria researchers themselves use to accept and reject sample material. If you were interested, it is only number 3 and 4 which are characterized by FTIR as plastics in the graphic here. Okay, we promised we're there. There's a moment in which suspected plastics become plastics themselves in the lab. So the spectrometer identifies materials by analyzing infrared absorption spectra. Connected software compares generated spectra from the sample with a known library of sample material. On the screen, we receive a list of possible matches between spectra generated, and any comparable spectra. So in this case, with this whitish film you can see on the laptop, it's been verified as some sort of polyethylene. The algorithm has returned a favorable result, which is a match here. Anything over 70% match is commonly accepted as being plastic in the community. What exact variant, we're not sure here, but that's enough for the researcher to go, okay, yes, check that on the spreadsheet, copy off the reading, take a photo, done. But it's not always so straightforward as that. A marine biologist that I was shadowing tells me a story of these bright blue and red fibers that are extracted from a marine organism. Running through the fibers, running through this process, FTIR confirms that they are cellulose-based, so natural, rather than synthetic in origin. These are also high percentage matches, over 70%. So it would have been appropriate for her to then just recognize these as not plastic, move on, go on to the next thing. Maybe write up as errors in analysis or visual identification. But given how abundant these blue and red fibers were, she thought that it was important to really dig into the results. So she initially hypothesized that maybe the enzymatic digestion process didn't clean the biological material enough, so it was giving an inaccurate reading. But however, more cleaning, same result, cellulose-based plastic. And so she decided to go for a second opinion from an analytical chemist who had a bit more expertise in vibrational microscopy. And after lots of toing and throwing on the spectral readouts, they determined that these fibers were possibly rayon or viscose, an alternative material to silk. These are cellulose-based, but synthetically regenerated and chemically altered polymers, meaning that while they're produced from a natural feedstock, they behave and function similar to a synthetic fiber. So questioning the data of this machine that purports to replace the human and offer automated analysis seems to render the very category of microplastic at risk. Are these cellulosic fibers natural or synthetic? Should they be counted as microplastics? We're at the end now. So hopefully this plastic has examined some of the complexities surrounding the sensing of microplastics. I've tried to do this through an empirical focus on so-called presence or absence studies. I've highlighted some of the key knowledge practices, methods, and instruments assembled in the lab required to make sense of microplastics and some of the challenges of transforming these sensory residues into evidence and data. But I also wondered if an account of microplastic that foregrounds these sensing practices might enable us to tell stories about the ubiquity of microplastics. I think there might be more to be learned from entering these laboratory spaces as a geographer. So we come to see that extracting sample material requires the researcher to pay intimate attention to the environmental milieu of the sample. So they are not just materials completely disconnected from their original environment, as Sebastian Rueta finds in his study of environmental toxicologists. Identifying materials involves more than visual attunements to microscopic matter and its situated geographies. In the commingling of sample and contamination in the laboratory space, researchers find practical, laborious, maybe a bit boring ways of keeping this ever-presence of plastics in check, even as they themselves are implicated in the experimental process. The dream of purifying the human and the social away through spectroscopy might need to be contextualized a little bit more carefully. But even in the analysis laboratory, where material separation and categorization are essential to the production of scientific knowledge, entanglements of plastics are not left outside laboratory walls. And maybe it is arguably this inattention to plastic materiality that has perpetuated the so-called bombshell errors in measuring microplastics in human samples, as analytical techniques treated as recipes to be reproduced in different settings and disciplines. So attention to these intersecting modes of sensing microplastics shift these material, chemical, bodily, social and ethical entanglements in the lab from being understood only as analytical challenges to be overcome in pursuit of maybe methodological standardization a faster throughput. These entanglements become the ontological starting point for this sort of lab work. So counterintuitively, to understand and respond to plastic entanglements, to return to the Wolf's provocation for STS scholars, you have to focus on these moments of disentanglements or separation, however improbable, momentary or partial. And in this way, perhaps these sensing practices provide yet another compelling reason for rejecting what Alexis Shotwell terms as a purity politics surrounding the solutionist discourse on plastic pollution that fundamentally undermine the realities of living with matter that will not go away. And what else is left to be said about the geography of microplastics beyond this statement of ubiquity then? I think the ubiquity of microplastics is almost taken for granted now, evoked as a sly and ironic indictment of the mess we live in. The problem presented by microplastics seems too vast and too small at the same time, but it turns out quantifying something that is everywhere is tricky, and generating data on microplastics is hard won. Focusing on how microplastic matter comes to matter is a way of spatializing ubiquity. It is this attention to the geographies of site, sample, and environment that enable microplastics to become sensible. But remember, microplastic science also has a geography. The processes, we've talked about processes focused on the UK, but the capacity to sense ubiquity is still unevenly distributed. As microplastic science as a field of research continues to expand and grow, new modes of sensing are constantly developed and come. And I hope this account demonstrates why it is important to critically engage with how microplastics become sensible, to recognize how these epistemic practices shape and constrain new forms of environmental awareness, responsibility, and regulation. Thank you so much for your time, and I really look forward to discussing this with you all. Thank you.